everyone, welcome back to my class. This is Rumana Ali. I hope you all are doing well. Strategies for enhancement of food production. Let us talk about the topic plant breeding for improved food quality. So more than 840 billion people are suffering from malnutrition. They are not getting enough amount of the nutrients and they do not have enough amount of the food to have and to maintain their health, and many of the persons are suffering from vitamin, mineral and protein deficiencies. We call this condition as hidden hunger. So there are many people who are suffering from this hidden hunger, who are starving and they are dying due to starvation. Some people are not getting the proper food, or they are not getting a balanced diet. Due to this, the risk of diseases increases, the lifespan of the people is getting decreases, and they are affecting with the mental disabilities. All such kind of changes can be seen due to the unimproved food quality, due to the improper supply of the food, or if we are not having balanced diet, then we need to face all these conditions. So diets lacking all such kind of micronutrients like iron, zinc, molybdenum. If the diet would not contain these micronutrients, then there are so many diseases which can be seen in the human body. The human can suffer from so many diseases if they are not getting a proper diet. So biofortification is a process by which the breeding of different varieties of the crops can be done to improve the food quality. Biofortification means it aims to improve the quality of the food, to increases the amount of the nutrients like vitamins, proteins, minerals, carbohydrates and fats. All such kind of components or the nutrients of the food can get increases by implementing a method called breeding. So by breeding the different crop varieties, we can improve the food quality. The main aim of the biofortification is to improve the oil content of the different foods. It also increases the vitamin content, and the main objective of the biofortification is to increases or to improve the mineral content of the different food products. So these are the main objectives of the biofortification. That means breeding to get the improved food items. To improve the quality of the food items, we are going for a process called breeding, that is nothing but biofortification. So by following the breeding method, we can grow different maize hybrids, we can grow the different hybrids of rice. In 2000, maize hybrids are produced, and these maize hybrids contain more amount of the amino acids like tryptophan and lysine when compared to the existing maize varieties or maize hybrids. So tryptophan and the lysine are the important amino acids that can be seen in a more or in a larger amount present in the maize hybrids when compared to the different maize crops. The next one is rice hybrids. Here we will take an example of golden rice. Golden rice is a hybrid that shows the presence of vitamin C. So golden rice is a hybrid which shows the higher amount of beta carotene present in it, and it is iron fortified rice. So this is how we can fulfill the nutritional requirements of the human population. The next hybrid is wheat. So when we grow the wheat hybrids, for example Atlas 66, it is a hybrid or it is a wheat hybrid that shows the presence of maximum amount of the protein. Maximum amount of the protein requirement can be fulfilled by having this Atlas 66 wheat hybrid. So this is how the plant breeding technique helps in improving the food quality. The food quality gets enhanced or it gets improved by the process called biofortification. Let us talk about the other topic, that is single cell protein, SCP. So we know that 25% of the human population is suffering from hunger and malnutrition. So one of the best alternative source of getting protein is through microbes. So microbes can be used as a source of protein on an industrial scale. Microbes like fungi, bacteria and algae, they are in use in an industrial scale for the production of protein. So these can be used as a source of protein, and as they are single cell microorganisms that are using in this production of proteins, we can term these protein as single cell protein. Here we will take one example, that is spirulina. Spirulina is a microbe that can be used as a source of protein, and it can be grown best by using the waste materials like molasses, straw or sewage, the waste water which is excreted out from the potato plants, that. So these all waste materials can be used to grow this microbe spirulina, and spirulina is the best source of protein. We can get more amount of the protein from this microbe spirulina. So by growing the spirulina, one benefit is it's a source of protein, and the other one is environmental pollution can be reduced. As we are utilizing all such kind of waste materials in growing spirulina, it also helps in decreasing or reducing the environmental pollution. This is how the soil and the water pollution gets reduces by using all such kind of waste material in order to grow the spirulina, which is a main or which is the best source of protein for human consumption. And it has been calculated that 250 kgs of cow can give 200 grams of protein, but the microbe like Methylotrophus, its biomass is 200 gram. Methylotrophus is a microorganism. So the 200 grams of this microorganism can give 250 tons of protein in the same time. So this is how we can understand that microorganism can be used as the best alternative source for the proteins. They are producing this much amount of the protein as their biomass is increasing rapidly, and also the growth of these microorganisms increases, so they can produce 250 tons of proteins. This is how the different microorganisms like fungi, bacteria and algae can be used as a source of protein. These microorganisms are best for the human consumption and also for the animal consumption. Let us talk about the other technique, that is tissue culture. In this type of technique, from any part of the plant we can regenerate a plant. So this is a technique in which we are going to take any part of the plant like root, leaf or stem, and from that part the regeneration of the plants takes place. Here we are taking any part of this plant. This is a main plant. If we are taking a piece of this leaf, then this is called as explant. From the explant, a new plant can be regenerated. This technique is called as tissue culture technique. In this technique, the explant is used, it is kept in a nutrient medium in the test tube, and sterile conditions are provided to regenerate the plant. Let us see what happens in the tissue culture actually. So the main thing is, here we are taking explant, that means any part of the plant, and the other thing that is needed is nutrient medium. This nutrient medium should be enriched with the inorganic salts, vitamins, minerals, amino acids, and growth regulators should also be present in the nutrient medium. This explant and the nutrient medium are important in the technique called tissue culture. So this nutrient medium, as it contains all such kind of inorganic salts and other substances like vitamins and minerals, then there is a chances of the growth of the microorganisms. To get rid of this problem or to inhibit the growth of the microorganisms in this nutrient medium, we are going to sterilize both explant and the nutrient medium in a sterilizer called autoclave. Autoclave is a steam sterilizer. In this sterilizer, sterilization takes place, and this sterilization takes place at 121 degrees centigrade for 15 minutes at 15 pounds of pressure. These are the conditions that needs to be maintained in the autoclave. The temperature should be 121 degrees centigrade, and the timings that are required for the sterilization in an autoclave is 15 minutes, and it should be done at 15 atmospheric pressure or 15 pounds pressure. After the sterilization of both explant and the nutrient medium, then the explant is inoculated in this nutrient medium, and this step is called as inoculation. After the explant is introduced into the nutrient medium, then this nutrient medium should be incubated for 3 to 4 weeks, and during this incubation period, the cell or the explant can undergoes the division. It divides rapidly within this 3 to 4 weeks, and it forms callus. Callus is proliferated and differentiated mass of cells. So, this callus is formed. After the formation of the callus, callus either grows into a plant directly or it undergoes a process called organogenesis. Organogenesis means development of the roots and the shoots. The shoots and the roots gets developed after some time, and this callus can be converted into roots and shoots, and as a result it gives a plantlet. This callus can undergo organogenesis process or it can undergo somatic embryogenesis process. In this somatic embryogenesis process, it results in the formation of somatic embryoid. The callus can be grown into somatic embryoid, and these embryoids, after the germination of the seeds, it gives plantlet. So, this callus, which is an undifferentiated mass of cell, it either forms a plantlet directly or it undergoes organogenesis, that means the development of the root and the shoot takes place, that gives a plantlet, or it undergoes a process called somatic embryogenesis that forms the somatic embryoids and it gives plantlet after the germination of the seeds. Here the embryoids are called as somatic embryoids because the sterile conditions are maintained in this tissue culture technique. The somatic cells are divided, so it is named as somatic embryogenesis or somatic embryoids. In this way, the tissue culture is a technique in which explant is inoculated into the nutrient medium. Nutrient medium contains all the plant growth regulators and the other substances. Plant growth regulators like auxins and cytokinins are also present in this nutrient medium that enables the explant to undergo differentiation or to undergo division. After the repeated division of this explant, it results in the formation of callus. This callus gives the plantlet. So, tissue culture is a technique which takes place within a shorter period of time. So, by this technique, we can produce a large number of plants within a shorter period of time and within a limited space. So, this technique can also be termed as micro propagation, and as the explant divides and form callus, this process or this capacity of the explant to undergo division in order to form callus is called as totipotency, and by this technique we can get soma clones. We can get the large number of plants by the somatic division, and it results in the formation of soma clones. So, this tissue culture is a process or it is a technique by which we can grow a large number of plants within a shorter period of time. And one more advantage of this tissue culture is to grow disease free plants by using the explant of infected one. Here we will take one example to understand this importance of the tissue culture. If a plant is infected some disease, then we will take a part or we will cut a part from its meristem. Meristem is a part of the plant where the cells are continuously dividing, and these cells are free from pathogens. So, when we take the meristematic cells from the infected plant and when we grow this explant or when we grow these meristematic cells in the nutrient medium, then it results in the production of the number of plants which will be free from diseases. So, in such a way we can develop disease free plants by this tissue culture technique. So, in this chapter we have discussed about the different strategies for the enhancement of food production, and that were plant breeding and tissue culture or micropropagation and hybridization or the production of the single cell protein. All such kind of techniques or the processes helps the farmers to increases their crop yield and to get better crop yield, and also to produces disease free or disease resistance, pest resistance crops. So, these are some of the techniques that can be employed by the farmers in order to get good yield and to increases their crop yield. That's all for today. I'll be back with a new chapter. Thank you.